Okay, let's take a look. So a lot of response for B. What is the name for the pumps that actively keep more sodium outside the cell, more potassium inside the cell? Absolutely correct. That is a sodium potassium pump, okay? And that's what we've spoken a lot about so far. So the answer here is B. Let's look at another. Okay, last few seconds. Right. Okay, overwhelming response for A. Neurons are generally more negatively charged on the inside than the outside of the cell. What is the name for this difference of charge when the cell is at rest? So really important, resting cells have this resting membrane potential, okay? So that's gonna be the resting membrane potential. So again, make sure you're keeping that terminology straight. It's gonna really help you really understand your questions, especially more complex questions that use these terms. So the answer was.
So does the membrane potential have to do with the charge of the ions, either inside or outside?
Yes, it does. So the membrane potential is the generalization of the separation of charge. Okay, when we speak about the resting membrane potential, it's pretty much the same idea, but it's really just describing that the cell, we're talking about a resting neuron here that's not being excited. Okay.
Okay, thank you.
Sure. Okay, and then let's look at one final question. All right, final few seconds. Okay. So why does the resting membrane potential tend to be closer to the equilibrium potential of potassium than that of sodium? And just to kind of further explain this here, remember we said that the equilibrium potential for potassium, it's a tongue twister, is -94. For sodium, it's +60. Now, as you can see, the resting membrane potential is -70. And so it's closer to potassium's equilibrium potential. So the question is asking, why is that? And so overwhelming response for C, which is correct, that is because of the differing permeabilities. The fact that the membrane is more permeable, 25 times more so than to potassium than it is to sodium, okay? So there's more free movement of potassium. Although it has weaker force, there's more opportunity for potassium to move. And so what the cell looks like at rest is going to closer mimic potassium's equilibrium potential just because it has more opportunities to move through the cell membrane. Okay. All right, so the correct answer here was C. Okay, so let's get into our next learning objective here. Let's kind of introduce this next topic. We're going to speak about properties of graded potentials. We're going to speak about the direction of change in a potential, the magnitude of change, right? How strong that force is. And then we'll speak about how we can start to sum those potentials using temporal and spatial summation, okay? And then later on, speak about how graded potentials can lead up to an action potential. So a graded potential is really a type of electrical signal. It is a small electrical signal. It's communicated over a very short distance. It's just a little blip in the membrane potential, okay? It can be a positive or a negative change. So it can be an increase in that value becoming more positive. It can be a decrease in that value, as we can see here, becoming more negative. And so it's a really small, it's not meaningful, it isn't sustained, it doesn't communicate over any length of a neuron. It's just a very small kind of blip in the membrane potential change that is not sustained, that is not meaningful. An action potential, on the other hand, is kind of the opposite of that. It is a large, sustained, meaningful membrane potential change. It's communicated over long distances. So if we think about the structure of our neurons, they're not just circular cells, although for simplicity's sake, we've been drawing them that way. They're actually these really long cells, like they have a soma that has receiving electrical inputs, and then they have a long axon that can travel very long distances. If we think about our lower extremities, the sciatic nerve, for example, spans the entire length of our limb. So these are the distances that an action potential can travel. And so it's a much more sustained, much more meaningful membrane potential change that has the ability to do work, okay?
Was graph B the example of action potential in that last slide?
On the graph here?
Yes.
So this is an action potential here. It's a positive membrane potential change. As you can see, it's much more, it's stronger in terms of its magnitude, right? This goes all the way up to positive 30, and then that is going to be able to be sustained, as we'll talk about later on.
So would action potential be like classified as like crossing the threshold in a positive?
Excellent. Yes,
Okay.
perfect, excellent. So once we hit that threshold value, which is negative 55, that is where it's sustained. Okay, it's not graded. A graded potential can have different valuations. It can go up to positive 40, excuse me, it can go up to negative 40. So we're starting at negative 70 here. It can go up to, let's say, negative 60, negative 65, right? It can have different gradations. When we get to negative 55, it must go up to positive 30. There's no gradation to an action potential. It's going to be an all or nothing phenomenon. So crossing that threshold value of negative 55 is going to definitely take us up to positive 30 and then back down to rest. Okay. All right. Hope that made sense there. So let's speak a little bit in the remaining minutes about the structure of a neuron and just describe some of these potentials. So let's start off looking at the basic structure of a neuron. It's got this soma, also called a cell body at the proximal end here. It's got this long projection centrally, which is the axon. And then it ends in this bulb, which is called the axon terminal. Okay. We've also got these projections that are coming out or sticking out of the soma called dendrites. And I'll label these off here in a minute. So we've got these projections, which are called dendrites. These are receiving input signals. We've got the central region here called the soma or the cell body. Okay. We've got the axon terminal toward the distal end. This is also called the terminal bouton, but only in muscle, right? Only if it is synapsing onto muscle. So I'm going to put this in here, put this here in brackets, the terminal bouton. specific to a synapse happening to muscle. Okay. And then the long projection in the center here is called the axon itself. Okay, and this can have varying lengths depending on the length of that nerve and how long it spans. Okay, we also want to think about an area of cell-to-cell communication. So because we have other neurons, other axon terminals of other adjacent neurons, synapsing onto the soma of this neuron, right? We have other neurons relaying their information here. We then have this region called the synapse, okay? So the synapse is this region where there's exchange or passing or communication of electrical information. And we'll talk a lot about the synapse coming up. So this is the synapse. And we're also wanting to label these neurons. This is going to be an important relationship that we'll go on to speak about. So because this is sending information from here to here, this can be labeled as the presynaptic neuron. And this larger neuron that we're focusing on is labeled as the postsynaptic neuron. Okay. Other important regions of the neuron is this central sort of neck region of the neuron. This is called the axon hillock. All right, this is the axon hillock. And we'll talk about why this is so important for action potentials. So if we think about graded potentials and their ability to sum, right? They're small membrane potential changes, but they help to get us closer to that threshold value, which would eventually result in an action potential. And so thinking about that, we can then relate different parts of the neuron to different types of potentials, okay? So the axon hillock is kind of where that distinction lies. So we have this separation here. Proximal to the separation, we have graded potentials. Right? Now, because these cannot be sustained, they're lost. So we lose that electrical activity through the cell. So if you think about positive and negative change, just like if you had electricity moving down a wire, if it were not properly insulated, that communication would not be sustained. And that is why graded potentials are not sustained. We dissipate that electrical activity. It's kind of watering down that electrical charge. And so they don't last for very long. Unless they get to that negative 55 threshold. Action potentials, on the other hand, do. And we'll talk about why they can be sustained and they can travel down the length of this neuron and be sustained. But they do have the ability to be sustained. But in terms of the structural distinction, they happen at the axon hillock and then they progress down the length of the axon. So here onward is going to be an action potential. All right, and I'll just kind of describe this here in brackets. It's going to be small, unsustained, because the electricity dissipates. Oops. Right, dissipates or dissolves as that travels further. Action potentials are going to be large, meaningful, membrane potential change that is sustained and communicates for long distances. Okay, and so basically what the axon hillock does is it's this region of integration. It's going to take all the graded potentials that are coming in. We've got graded potentials coming in here. We've got graded potentials coming in here. We've got graded potentials coming in here. And at this point, we're going to really analyze all of these graded potentials, the cell is going to decipher, have we succeeded that threshold qualification? Are these minimal graded potentials, enough in terms of their magnitude to get us to that -55 threshold? And if it does, then we're going to whip an action potential. But if it doesn't, then they're simply going to dissipate, okay? So this is where that -55 threshold really comes into play. This determining if we meet that qualification to then initiate an action potential.